Thank you very much. Can you hear me? Oh,
Yes, we can hear
sorry.
you.
We can, yeah.
Yes, yes, we tested. I know. So let me share the screen first. Slideshow. Okay. So thank you very much for the kind introduction, and thank you very much for the opportunity to speak at this long-established prehistoric society lecture series. My name is Shinya Shoda, the head of International Cooperation Section, NARA, National Research Institute for Cultural Properties, Japan. I am also affiliated with University of York and Sainsbury Institute for the Study of Japanese Arts and Cultures. Both are located in the UK. Let me start from self-introduction. I am both a field and lab archaeologist whose research field is mainly Northeast Asia. Born and trained as an archaeologist in Japan, did PhD in Korea, and got a job in Japan. I had been conducting my research project in BioArc, University of York, from 2014 to 2016 as a Marie Curie Fellow, as introduced, where they were integrating biochemical analytical methods to answer archaeological questions. My research topics are not easy to explain systematically as I do whatever I want to understand, regardless of time and place, but they can probably be listed in this way, at least partly. Transition from origins and motivations for pottery innovations, transition from hunter-gatherer fishers societies to agricultural societies, origins and motivations for pottery innovations, dietary and cuisine culture in the past in different periods and places, as well as its deep time background. And finally, integration of humanity and natural scientific method for archaeological research. So that's me. Here is the outline of my talk today. Assuming that many of you are not familiar with organic residue analysis, I will start from a brief introduction of the methodology that I use. The main part is divided into two. One is on the beginning of pottery in the different part of East Asia, and another is on the millet dispersal in Central Asia. Let's start the introduction part. What is organic residue analysis of pottery? If you are interested in Japanese archaeology, this is one of the most frequently introduced objects from the Jomon period, prehistoric Japan, which is called flame pottery or kaen toki. Now, as a designated national treasure, even experts are no longer allowed to touch it except in limited occasions, including myself. But actually, just before the registration as a national treasure, I was fortunate to have an opportunity to observe this pottery when I was undergrad student. When I looked inside, which is probably impossible today, but can you see this? Brackish bands of carbonized deposit. Apparently, this pot was used for cooking something rather than an artistic object as it is today. So I want to understand in what context and what kind of a food they were cooking using this pottery. This is a typical example of our samples, potsherds without any beautiful decorations. But for me, this is as valuable as national treasures. Hopefully, you will understand it after my talk. From potsherd like this, ceramic powder and or charred surface deposits are sampled. But how can we draw culinary evidence from here? Before getting into details, I just want to show this YouTube video as an introduction to this method. It is said that seeing is believing. So I'm gonna stop this for this moment and share another file. Done? Oh yeah. Okay. So let's start again. Oh, sorry. How is the sound? Okay.
こんにちは。奈良文化財研究所の正田信也です。今日お話しするのは土器についてです。これまで土器の研究といえば文様の付け方や形、大きさ、あるいは作り方などを調べて、それがいつ頃どこで作られたものなのかを研究するのが一般的でした。でも今ここで私たちが取り組んでいる研究のやり方は考古生化学と呼ばれる最新の科学技術を用いた研究のやり方です。例えばその一つに土器残存質分析と呼ばれる研究方法があります。この研究方法を用いることで大昔の人々が何のために土器を発明したのかといったことを科学的に研究することができます。土器の表面や内部には目に見えないほどの小さな穴が無数にあります。この小さな穴に土器が使われた時のさまざまな物質が残されることが分かっています。例えば動物の脂肪や植物の油といった脂質と呼ばれるものが典型的なものです。土器にどのような脂質が残されているのかを分析することによってこの土器がどのように使われたのかを明らかにすることができます。まずは調べようと思う土器の表面をドリルで薄く削っていきます。土器が使われなくなってから付着したさまざまな物質を取り除くためです。その上でドリルの先端をきれいなものに取り替えて土器の内部を削り分析に使う試料を採取していきます。この試料にまずメタノールと硫酸を加えて化学反応を起こし機械にかけてさらに4時間加熱します。それを遠心分離機にかけます。そうすると土器の粉の中に含まれていた成分が溶液の中に移っていきます。そこにヘキサンという薬品を加えて攪拌機にかけます。こうすることで溶液の中に含まれていた脂質を透明なヘキサンの中に移すことができるのです。最後にヘキサンの溶液に窒素ガスを吹きつけて濃縮していきます。これを繰り返すことで分析するのに十分な濃度の試料を得ることができるのです。こうして抽出した脂質をいよいよ分析します。ガスクロマトグラフという機械を用います。こちらが佐賀県の東妙遺跡から出土したおよそ8000年前の土器を分析した結果です。3箇所のピークのまとまりが見られます。これはアルキルフェニルアルカン酸といって水中の生き物、例えば海や川の魚に多く含まれる脂質です。こうした分析を積み重ねていくことで興味深いことが分かってきました。日本列島は世界で最も古くから土器が使われている地域で1万5000年以上前に遡りますが、初期の土器からは魚介類に含まれる脂質が多く検出されました。このことから日本の土器は水産資源を調理するために生まれた可能性が浮かび上がってきました。同様の結果はロシアから朝鮮半島にかけての同じ時期の土器でも出ています。一方、東アジアよりも遅く9000年前に土器を使い始めた西アジアでは状況が少し違います。イギリスの研究グループの分析によると家畜の脂肪や乳製品に特有の脂質が検出されているのです。これらのことから西アジアでは土器は牛、羊、豚などの肉の調理や乳製品の加工用に発達したと考えられるようになってきました。現在私たちの研究室ではカザフスタン西部をフィールドに土器残存脂質分析を進めています。それはこの地域にはタクサイ古墳というとても興味深い遺跡があるからです。この古墳は黄金の装飾品をまとった 20代の女性が埋葬されていたことで有名ですが、この遺跡から出土した土器を私たちは調査しました。その土器から検出されたのはミリアシンと呼ばれる化合物でした。ミリアシンというのは雑穀のキビの実に特徴的に見られる物質です。この土器はキビを煮炊きするのに用いられたということがわかります。さらに興味深いことがあります。この遺跡では多くの人骨が見つかっていて、その骨の研究も進められています。するとこの遺跡の人々は子供の頃にキビなどを多く食べるものの、大人になると食べなくなるということが分かってきました。つまりこの土器が子供の食事の煮炊きに使われていた可能性、そしてその土器を墓に埋葬する習慣があった可能性が浮かび上がってきました。土器の形や文様の分析だけでは見えてこない人生のエピソードが伺えるようになってくるのです。土器残存資質分析はこのようにいろいろな可能性を秘めた非常に魅力的な分野です。皆さんも私たちと一緒に研究しませんか？
Did we come back to the slides altogether? Yeah. Okay, so let's move on. This shows a little more detail than in the video, but still simplified protocol. We need to drill pots to make it into powder for lipid extraction. Lipids are extracted and derivatized to be analyzed in gas chromatography, or simply GC. Two different kinds of devices attached to GC are used for two different approaches for identification. One is GCMS for biomarker identification. The other is GCCILMS for the measurement of compound-specific carbon isotope ratio. I will add short explain for each. The word biomarker is first used in geochemistry, as the word means organic compounds preserved in the geological record and are characterized by specific carbon skeleton that might be related to their biological source. Later, in archaeology, the term biomarker is mainly used for natural sources, although there is no strict definition. Sometimes anthropogenic, degradation, and migration or contamination markers also can be detected. Compound-specific isotope ratio doesn't change through time, as it is focusing on single compounds. Palmitic acid and stearic acid are two major fatty acids that are frequently observed in archaeological record, and by measuring stable carbon isotope ratio of these individual compounds combined with archaeological information, sometimes we can identify the origins of lipids in archaeological remains. This is the very early study using this method by Richard Eberschet, which successfully identified the non-luminant fat and luminant fat from different types of pottery. It was possible because luminant, such as cattle, sheep, and goat, and non-luminant, such as pig, have different carbon source to produce same fatty acid because of their different digestive systems. It says the associations of different fats with different vessel types might be culturally significant. Hopefully, you're somehow familiar with lipid residual analysis within just 10 minutes. Too short, sorry. But now, I show you how we interpret the data based on my own work. As you know, results or data never give us the answer, so we need to interpret the data. This is typical case for the identification of marine origin. Left is the GC chromatogram, which can be interpreted as aquatic, based on series of diagnostic compounds that are so-called aquatic biomarkers. But only by this, we don't know whether they are from marine or freshwater, and here we can measure their individual fatty acids carbon isotope ratio. As shown on the right hand, the product safely came into the marine range, so accordingly, this sample is highly probably originated from marine organisms. Another famous biomarker is meliacin, which is the diagnostic compound for the grain of broomcorn millet, but is absent in many other commonly cultivated species. This is also from my study in Korea, and on the right hand, the plot with M character shows the presence of meliacin, while they are showing different isotopic values, sometimes outside of C4 plant reference range. This indicates that the pottery was not only used for cooking millet, but also used for cooking other ingredients. In a case of carbonized deposit, millet was highly likely to be cooked with other ingredients, while in case of ceramic powder, millet and other ingredients were cooked in the same vessel in its life history. Thus, we don't know if it was at the same cooking event or not. Now let's move on to the exciting case studies. The first one is on East Asia, looking at very early pottery in different areas. Oh, doesn't work well. Hey. Just a moment, something is going wrong. Come on, why? Okay, it's working finally. So this is a map by Peter Jordan and his colleagues, who is an enthusiastic researcher of early pottery. As shown, recent achievement in African archaeology show that different pottery origin than East Asia is now being discussed. But today's talk will focus on the east side of the continent, where there are mainly three core areas of earliest pottery: Japan, Russian Far East, and South China, highlighted by black color, clearly showing that this area is definitely a key area to understand the innovation of pottery technology. Not in many parts of the world, people began to use pottery as early as late Pleistocene, like here. Also, at a glance, you will be aware of the orange color, which means that around 8,000 BP, pottery use was largely expanded into larger area of the world. As I indicated just before, there is an expansion period of pottery use, which is far later than the beginning of it. This S-curve model explains it well. Gibbs and Jordan have divided the development of pottery into 4 stages: experimentation, intensification, integration, and dispersal. So not only the beginning, but also the intensification stage is important to understand the innovation process of pottery. Okay, the oldest pot from Japan, which dates back to 16 to 14,000 years ago. They are all fragments, but the important point is they have some brackish adhered surface deposit on the interior and exterior of them. They were highly probably used for cooking something, as previously had been assumed. In old days, there is no direct method to examine what they cooked in prehistoric pottery. One assumed that as nuts, because some of Japanese nuts require removal of toxic components such as saponin and tannin. This hypothesis fit in the assumption that pottery began to be used in the early Holocene when they have broadleaf tree forests around. But this is not true anymore because the latest dating of early pottery tells us that pottery was invented in late Pleistocene, which is the Ice Age. The vegetation consisted mainly of conifers. If not nuts, what was cooked? In 2013, a new research was published in Nature. This paper by Craig et al. Revealed that the preparation of aquatic resources, particularly marine foodstuffs, was the dominant mode of pottery use in Japanese early pottery. This conclusion was led by the result of lipid residue analysis pottery. This was groundbreaking study for Japanese archaeology, and this is the very moment that I got interested in this methodology. This result is also interesting to British scholars as the pottery use is totally different than that of British Neolithic that was mainly for ruminant animal, meat, fat, or milk. Interestingly, Craig and his team also revealed high aquatic resources contribution in the early Neolithic in Northern Europe. Based on these studies, this was a very simple imagination map in my brain in 2013. Fortunately, Professor Oliver Craig visited Japan right after the Nature publication, and we met each other to discuss collaborative research on East Asian early pottery, and double fortunately, the application for Marie Curie Fellowship was accepted. On this background, I started the project named Ponte, Pottery Innovation and Transition in East Asia, Bridging Expertise Across Continents with Oliver. The aim is, of course, the application of pottery repeat analysis on early pottery in East Asia. BioArc at the University of York provided the ideal environment, not only the facilities for archaeochemical analysis, but also a friendly environment surrounded by many colleagues with similar interests. We helped each other and we were able to acquire a variety of knowledge and skills in a very short time. This project was focusing on not only early pottery in Japan, but also that of surrounding area, such as Russian Far East, Korea, and South China, as well as Taiwan. 399 samples have been processed for the analysis by myself. Also, simultaneously, AHRC project named the Innovation and Development of Pottery in East Asia, led by Oliver Craig, is ongoing in the same place, which share the research method and focus with Ponte. Hundreds of German pottery were analyzed in this project, in addition to my own project. This study focused on a long-lasting site in the middle part of Japan. You may see the location and time period at the right bottom of each site. The data obtained from Torihama site showed high percentage of aquatic biomarker detection through time from the incipient to early Jomon for about 9,000 years. Closed plot in these graphs have aquatic biomarkers. Although the shift from marine to freshwater can be observed, fundamentally, they kept on cooking aquatic organisms as long-time tradition in spite of drastic climatic change during 9,000 years. Other archaeological evidence, such as botanical and faunal remains, show that they have a broad range of food resources around them. Contrastingly, pottery seems to be used for limited purpose. This result is quite unexpected because we expected change of pottery use according to the environmental changes from Pleistocene to Holocene. Another possibility is that this is just a site-specific phenomenon. To test if there are any sampling bias, more than 800 potsherds from all over Japan were analyzed to test the hypothesis that the early pottery in Japan was dominantly used for cooking fish, as was concluded in the previous papers. As a result, both incipient and initial Jomon pottery were dominantly used for processing aquatic resources in spite of different time and different location of the sites. This indicates the existence of strong social tradition for pottery use. Across the strait, this is the Sejuk-Cheonmyeon site on the southeast coast of Korea. From this site, nuts and wild fruits, as well as variety of animal bones were recovered. Pottery lipid residues analysis showed again the high concentration to aquatic resources, especially to marine resources. In this site, We have even discussed the possibility of rendering oil in pot based on the almost identical isotopic values between bulk and specific compounds. We need to think about why they processed aquatic or marine resources intensively and rendering oil to get high calorie and stable storable food is one of the possibilities. One can possibly doubt that this tendency is because these sites are shell-midden. That was the reason why I chose this site, the Chukbyonni site, for the next analysis. This site is non-shell-midden site and divided into two localities. Locality Chunno 3 seemed to have some superiority in location compared to locality 1568. The results are basically similar to other Holocene sites in Northeast Asia, which is highly aquatic, but there's difference between these localities. The former with some superiority has concentration in the terrestrial area without aquatic biomarkers, and these pots are so-called red burnished pottery that are elaborated ones. The result shows an intra-site difference of pottery use, and it is interesting to know that some exceptional use of pot in this period are combined with elaborated pottery. But if we integrate the result from these two Korean sites, pottery were dominantly used for marine resources as shown as the isotopic values, which is similar pattern to the German pottery. Now again, the map in my brain in 2014. Japan and Korea are recognized as aquatic early pottery zone. Now, how about the continental early pottery in Russian Far East? Fortunately, I was able to obtain some of the oldest pottery shells in the world for the analysis from the middle and lower Amur River basin. Again, the results showed the significant contribution from fish. In this case, variety of fish, including marine, salmonid, and freshwater fish, seem to be cooked in pot. If we compare this result with incipient initial German pottery, at a glance, they look like similar. Again, aquatic story. However, you don't have to be disappointed. A closer look at the middle Amur and lower Amur River basins reveal interesting patterns. In the middle Amur River basin, highlighted by cold colors, pottery was mainly used for ruminant fat processing, while in the lower Amur River basin, highlighted by warm colors, pottery was used for cooking fish with a particularly high proportion of salmonid fish at the Goncharka site. These results indicate that differences in pottery production traditions correspond to differences in the use of pottery, and therefore that the motivations for pottery invention differed from region to region in the very early stages of pottery use in human history. Now, let us move on to the South of East Asia. Unfortunately, I have not yet had any chance to approach the earliest pottery in southern China, dating back to 20,000 BP. But very interesting results were obtained from the Tianluoshan site around 8,000 BP, that is expansion period of pottery technology to larger area of the Eurasian continent. This site is well-known waterlogged site where they have excavated a variety of organic remains, including wooden and bone tools, plant seasoned fruits, and animal bones. Enormous amount of pottery was excavated, and I was lucky to be allowed to take any of these pottery for the analysis, thanks to the excavation director. So I decided to focus on full boiling pots, as well as char deposit on the bottom pottery. The result differed significantly from any previous results. Levoglucosan, a compound formed from the pyrolysis of starch and cellulose, was detected in many samples, as was phytosterol from plant sources. Also, very depleted carbon isotope values of specific fatty acids strongly indicates the significant contribution of C3 plants such as rice, water chestnuts, or foxnuts that are abundant in this site. Additionally, the trace of beeswax was detected from a char deposit on the interior of bottom sherd. This sample was directly dated and revealed to be the earliest evidence of beeswax, thus highly likely honey, in East Asia. Let's sum up the part one. Pottery use had been strongly oriented for processing aquatic resources in Northeast Asia, despite long-term environmental change. However, the motivation of pottery invention or disperse could be diverse according to the environment in different areas, such as in Russian forest and southern China. Now, part two. Moving to the Central Asia. Here, I focus on millet, but why millet matters? Some of you know about this. The last year was International Year of Millets. The objective of this is to raise awareness of and direct policy attention to the socio-cultural, nutritional, and environmental benefits of millet cultivation. Indeed, in prehistoric northern Eurasia, millet is one of the most important crops. Many of you may know this eye-catching phrase, prehistoric food globalization, by Martin Jones and his colleagues. Indeed, 3rd to 2nd millennium BCE and following centuries witnessed the dynamic interaction between East and Western Eurasia in terms of domesticated plant and animals, as well as variety of their use. This movement was called as prehistoric food globalization. Two species of millet, broomcorn and foxtail millets, were originated in northeastern China and spread out in all directions as highlighted in red color. Just simply, I have drawn a cross-sectional map of the topography of the route along which the wheat crops were transmitted from west to east and the millet from east to west. Instinctively, we can see that this is a big deal. Not only would humans, but the cultivated plants and livestock that moved with them would also have to adopt to the dramatic environmental changes. And this makes humans to seek different kind of cultural adaptations in different settings, including their diet and culinary practices. Studies that have analyzed stable isotopes of collagen in human bones have revealed some very interesting trends. The upper left-hand figure shows that the trends in plant intake reflected in human bones from the Yellow River Basin to Europe are clearly different, with the boundary between Gansu and Xinjiang area in Western China. In the East, the intake of millet with their enriched carbon isotope value is more pronounced, while in the West, on the contrary, the intake of wheat or barley, C3 plants, with its depleted carbon isotope value is observed. As for the time period between 2500 BCE and 1500 BCE, there was a dynamic mixture of Eastern and Western tradition. This map shows the situation in 2500 BCE, and this shows 1500 BCE. As you see, one of the hotspots of this dynamic mixture happened in today's Kazakhstan. So I thought it would be useful to track millet consumption in Kazakhstan. After finishing my Marie Curie at York in 2016, I came back to my institute and started preparing my lab. It took a few years to get ready by setting up equipment and hire postdoc, researcher, and technicians, but now set up. At the same time, I have been appointed as head of International Cooperation Section, whose aim is promoting international collaboration and knowledge transfer to the overseas countries. Very fortunately, I could start this project in Kazakhstan, funded by Japanese Agency of Cultural Affairs called KOPIR, meaning bridge in Kazakh language. As you might be aware, I just mimicked my project in York called Ponte, meaning bridge in Italian. I started with the Taksai site, which is famous for the golden findings associated young individuals, but for me, sorry, something going wrong. So you know about this site because our video shows the result already. So this site is famous for the golden ornament and it's a young lady, but more than golden object, the pottery is even more valuable for me. The sample was taken from Kurgan 2 in the Taksai 2 site, where they excavated several barrier structures. A char deposit was sampled from this offering pottery. 2 biomarkers were detected in this sample. One is miriacin, which is the one in question from earlier, and the other is this is difficult to pronounce actually, alkanoic acid, which is produced when heated at more than 200 degrees for 5 hours. We therefore conclude that the millet was cooked in this pot. The compound-specific carbon isotope tells us additional story about the cooking. The value shows the clear contribution of ruminant adipose strongly indicated that millet was cooked with ruminant meat or fat, such as cattle, sheep, goat, or saiga. This reminds me of very impressive spring festivity in Kazakhstan, to where my Kazakh colleagues kindly invited our team. The right bottom is called Nauruz Kuzhe, the mixture of grains and meat, as well as milk. Of course, no daily signals was confirmed from this sample. It might be porridge like cooking something similar to this. The archaeological context of this site helps us further interpret the culinary practice in this site. According to the study by Ananievskaya and colleagues, it is clearly highlighted that there is clear division of millet intake according to their age. Individuals under 12 years old showed enriched isotopic values, most likely because of C4 plant intake, while much depleted values in the adults, individuals, indicating C3 dominant diet. So the porridge-like cooking we found might be feeding children. Why? Is there any difference in food values? Any allergic problem? Just only one result arose many new questions, and this made me fall in love with Central Asian archaeology. We continued and expanded our research into larger scale, conducting repeat residue analysis pottery from different areas of Kazakhstan, focusing on Iron Age period. On the left, I show the frequency of miriacin detection in different sites, showing much higher ratio in the southeast Kazakhstan. This is well corresponded to the C4 signals reflected in the human bone collagen studied by Ananievskaya and colleagues, shown on the right. This somehow makes good sense because this site is located on the very dispersal route of millet, which is called Inner Asian Mountain Corridor. Furthermore, the compound-specific isotope ratio shed new light on the variety of cooking among different areas and sites. Even within Kazakhstan, these 3 sites show clear difference when we pick up the sample with miriacin. The black curves with dots are the model mixing curves based on the calculation of isotope values of mixed ingredients of millet with other sources such as C3 plants. C3 plants fed non-ruminant animals were ruminant animal adipose, as well as milk of C3 or both C3, C4 fed animals in different proportions, different from each other by 10%. In Akpan site, millet is associated with ruminant or non-ruminant adipose. In Katartube, with C3 fed ruminant daily. In Kuzugurik, C3 and C4 fed ruminant daily. If you add the data from Korea, Korean settlement sites, the difference is even clearer. This is mostly to a mixture between C4 and C3 plants such as rice, as well as non-dominant animals and fish. I just put these pictures to show modern analogs, somehow corresponding to the prehistoric situation. You might think it is nothing very exciting to find different culinary practices in different sites or areas, or different types of sites such as settlement, cemetery, as well as sanctuaries. But if we take these two sites as example, these are all called sanctuaries in Kazakh archaeology, next to each other by 70 kilometers in the same period. This result is very inspiring to further research on the difference of the context in that the pottery was used, as well as the activities or function of the monumental buildings in both sites. Now, 45 minutes, okay. It's time to summarize the second part. Organic residue analysis can trace not only the millet dispersal, but potentially the frequency of its use. Second, millet might be cooked in pottery for preparing food for children, ? Third, regional diversity in millet cooking were observed, possibly related to the meaning of the places. So that's it. Thank you very much for kindly listening to my talk.